टॉपिक है वो है बैक्टीरिया। उम्मीद है आप सब हैरियत से होंगे। चलिए आज हम फटाफट चलते हैं अपनी टॉपिक्स की तरफ। Bacteriology. Bacteriology is the branch and especially of biology that studies the morphology, ecology, genetics, and biochemistry of bacteria as well as many other aspects related to them. Bacteria. What is bacteria? A prokaryotic and single-cell microorganism, small in size, don't have nuclear membranes, and also lack membrane-bonded organelles. Can be able to see by using microscope. यह है बैक्टीरिया की डायग्राम। कैप्सूल, सेल वॉल, प्लाज्मा मेम्ब्रेन, साइटोप्लाज्म। Bacteria are found in every habit on Earth, like growing in soil, acidic hot springs, and deep earth crust. Bacteria cells are 10 times smaller than the eukaryotic cell and are typically 0.5 to 5.0 micrometers in length. There are 14 million bacterial cells in 1 gram of soil and millions in 1 millimeter of water. Structure of bacteria: capsule, cell envelope, protective covering made of polysaccharides protect it from phagocytosis by large microorganisms. The cell envelope is made up of 2 to 3 layers: the interior cytoplasmic membrane, the cell wall, and its some species of bacteria, and outer capsule. Cell wall, composed of polysaccharides, give the cell its shape and surrounds the cytoplasmic membrane. Protection: cytoplasm helps in the cell growth, metabolism, and also in replication. सिलेक्शन ऑफ बैक्टीरियल सेल्स ये डायग्राम है। सबसे पहले डिस्कस किया इन फ्लोडिंग ऑफ प्लाज्मा मेम्ब्रेन, कैप्सूल, सेल वॉल, डीएनए coiled into nucleoid, basal bodies, flagella, ribosomes, cytoplasm, plasma membrane, cytoplasmic inclusion, pilli. आगे हम डिस्कस करेंगे पिलाए। फ्लैजिला कैप्सूल को हम बड़ी डिस्कस के साथ एक्सप्लेन करेंगे। Cytoplasmic membrane, layer of phospholipids and proteins, regulate the flow of materials in and out of the cells. Flagella, hair-like structure. Flagella का hair-like structure होता है. Provide means of locomotion. The flagella beat in a propeller-like motion to help the bacterium move towards nutrition away from toxic chemicals. Nucleus, region where the chromosomal DNA is located. It is not a membrane-bound nucleus. Most bacteria have a single circular chromosome that is responsible for replication, although a few species do have 2 or more. Pilli. Many species of bacteria have pilli, singular pillars, smaller hair-like projection emerging from the outside cell surface. These outgrowths assist the bacteria in attaching to other cells and surfaces such as teeth, intestines. Coccus. A cocci is an bacterium or a trichome that has a spherical, ovoid, or generally round shape. Arrangement: dipcocci are pairs of cocci, Streptococcus pneumonia. Staphylococcus are irregular shape like cluster of cocci. Staphylococcus aureus tetrads, a cluster of 4 cocci arranged within the same plant. Micrococcus sircinia is a genus of bacteria that are found in cuboidal arrangement of 8 cocci. Sircinia ventricula. Streptococci are chain of cocci. Streptococci folians. Streptococci ये मोतियों की तरह होती है, इसकी चेन जैसे आप देख सकते हैं। आगे हम डिस्कस किया है अरेंजमेंट कोकाई, कोकस, डिप्लोकोकस, स्टेप्लोकोकस एंड स्ट्रेप्टोकोकाई, सर्किना, इनसे सर्किनियस बोलते हैं। टेट्राड, ये इसके डायग्राम्स हैं। ये बॉन्डिंग की आपस में ये ओवरलैप हुए हैं एक दूसरे के साथ। अब हम डिस्कस करते हैं बेसिलस. बेसिलस is a genus of gram-positive rod-shaped bacteria. Bacillus shape can be either obligate aerobic or facultative anaerobic bacillus. Obligata aerobe depend on oxygen. Facultative anaerobe have ability to grow in absence of oxygen. जिसकी दो टाइप्स हैं बेसिलस की, obligata anaerobe and facultative anaerobe. बेसिलस. बेसिलस की डायग्राम है ये. बेसिलाई. कोकोबेसिलस, बेसिलस, डिप्लोबेसिलस, पैलियोसाइट्स, स्ट्रिप्लोबैसिलस, Spirillum. Spiral bacteria are the helical shaped bacteria. They have two types, Spirillum sporocheete. On the base of cell wall, gram-positive bacteria, gram-negative bacteria. Thin cell wall in gram-stance test give positive results, stance the cell wall purple. Gram-negative bacteria, thin cell wall in gram-stance test give negative results, stand the cell wall pink. Difference, what is the difference between gram-positive and gram-negative bacteria? On the basis of nutrition, autotrophic bacteria, hitrotrophic bacteria. Food is synthesized from simple inorganic raw materials, carbon dioxide and water, CO2 and H2O. Green pigment is necessary. Food is prepared during daytimes. Example are green plants or some bacteria. Heterotrophic bacteria. Food is obtained directly or indirectly from autotrophs. No pigment is necessary for nutrition. Food can be prepared at all the time. Example are animals and fungi. Difference between autotrophic and heterotrophic bacteria. Let you discuss a type of heterotrophic. Saprophytic bacteria obtain their food from death and decay matter. Decomposer have on enzyme system. Parasitic bacteria lock on enzyme system depends upon enzyme of host living for food pathogens. Symbiotic bacteria symbiotic relationship with host. On the base of flagella, types of flagella arrangement. Let's you see discuss arrangement of flagella. Polar monotrichous single flagella at one pole. Lapotracheous stuff of flagella at one pole. Amphitracheous flagella at both poles. Patricheous flagella all over. Amphilophytrocheous stuff of flagella at both ends. Let you see diagram of arrangement of flagella. Method of replication: sexual reproduction and asexual reproduction. Asexual reproduction in asexual reproduction, single organism produce genetically identical offspring. Types: endosperm formation, vegetative reproduction. Endosperm formation, resulting spore found in gram-positive bacteria during formation protoplasm, construction around the chromosomes, and a hard resistant wall secreted around the rest of the bacterial cell degenerator. Let's discuss formation, spore formation, bacterium. Bacterium, endosperm, germination of endosperm, parental cell breaks. Vegetative reproduction, beneficiation, budding, fermentation. Beneficiation, the division of a bacterial cell, parent cell enlarge and duplication in DNA. Septum formation divides the cell into two separate chambers. Complete division result into identical cells. Let you see diagram of binary fission. Next topic is budding. Smaller protobryonia bud develops at one end of bacteria. Genome replication occurs and one copy of genome gets into the bud. Bud enlarge and evolutionarily separate from the parent cell. Bud belongs to form on the parent cell. Nuclear copies and divides the bud receive a copy. Bud now become a separate daughter cell. Budding produce change of cells. Budding produce change of cells. At last, bud produce